Okay, hello everyone again. Moving on to from the last lecture talking about amino acids and proteins. This lecture is going to dig in a little bit more about proteins, how you purify proteins, how you measure proteins, some analytical work you can do with proteins, and some more information about why proteins are important in the context of medicinal chemistry. Again, my email is shown here on the slide. Go ahead and email me with any questions you have about the lectures. So by way of an outline for this lecture here, just going to introduce you to protein purification, introduce you to analysis of proteins, some example proteins, and then I have a study guide that covers this lecture as well as the previous lecture. Protein purification and characterization of proteins. So when you're talking about biochemistry, these investigations a lot of times require pure protein components. So your cell contains all kinds of different substances between proteins, nucleic acids, lipids, sugars, and a lot of these biomolecules have similar physical and chemical properties. When we want to look at proteins, we got to get rid of a lot of those things. And the biomolecules, some of these may be unstable or they may be present in very small amounts. So we need to purify proteins to characterize them, whether that's for structural studies, protein-protein interaction studies, or if we want to develop drugs or drug targets or some sort of diagnostic. A lot of times we need to purify these proteins. So I'm going to talk through the main ways that people do this. So these impurities that are with our proteins of interest can either be due to the process, due to processes, or the product themselves. And so the processes, for example, can be from the other host cell proteins that we talked about. For example, if you're expressing a protein in E. coli, your protein of interest, you got to purify that away from the other proteins in the E. coli background. There can be proteolytic enzymes that can eat your protein, essentially digest your protein. You need to get rid of those. Glycolytic enzymes, there's endotoxins, lipids, again, DNA, even viruses in some of the preps, lipids, all these different cell debris, as I mentioned. Even the cell culture reagents that we may have used, the chemical additives, the detergents that you may have in there for solubilizing your protein. So all these things can be process related that you need to purify your protein away from. The product itself, the protein itself, can form dimers, multimers sometimes, can form aggregates, so you need to be worried about that. Sometimes they can misfold. You can get deamidation products. You can get other chemistry occurring like oxidation of methionine. You can also have some heterogeneity in your product, so some portions of your protein may be post-translationally modified. The enzyme can get degraded, as we've talked about. So there's all these challenges that you have to deal with when you're purifying a protein. So there's some general strategies that people use, and I'm going to talk through some of these, but all of them exploit some sort of characteristic of the protein. So one characteristic you can exploit is the solubility. So you can use the either procedure of salting in or salting out, for example, and we'll talk about that. You can exploit ionic charge. Last lecture I talked about the charges of amino acids, and you can use something called ion exchange chromatography, where you bind to, you use either positively charged or negatively charged resins to bind to your protein of interest. Electrophoresis is a technique where you can separate proteins, and I'll talk about that. Isoelectric focusing exploits the PI of the protein to separate proteins for analysis. You can also exploit the polarity of the protein by using various chromatographies such as absorption chromatography, paper chromatography, reverse phase or hydrophobic chromatography, and I'll talk through what those are. One of the most basic ways is just by the size of your protein. So you can separate out macromolecules from small molecules, even from different sized proteins. You can do that by a couple different techniques, dialysis or ultrafiltration, and I'll talk through what these are. Electrophoresis, in addition to using charge, it also separates based on size. And then gel filtration or size exclusion chromatography, I'll talk through what that is. That's a technique that is used a lot in protein purification. You can also use very specific things about the protein if it's known to bind to a certain ligand or bind to a certain other protein, for example. You can use that specificity for affinity chromatography, and this is exploited a lot in protein purification because it is very specific to your protein of interest. So if we talk through a couple of these, first, solubility-based purification. So essentially what you do here is you adjust the solution to just above or just below the point to which your protein of interest is solubilized. So for example, maybe a little bit hard to see here, but down in this diagram here, if you have a solution of your protein, just kind of the light triangles here, or light gray circles here, with the other darker colored circles and the white colored circles, you can essentially change the ionic strength by adding salt, changing the polarity, or changing the pH or the temperature to basically crash out other proteins in your solution, shown by the black dots here, whereas your protein and possibly some other contaminants still stay in solution. Then you can just separate these by centrifugation, pull off the supernatant, and then you have a little bit purer prep than what you started with. So you can do it in either way. You can salt in or salt out. Salting in is just where your protein solubility increases with increasing ionic strength, and this is due to shielding of protein charges or charges on your protein. The other way to think about it, the other way to do it is if your protein solubility decreases with ionic strength, so you increase the ionic strength of your solution and your protein crashes out of solution. And this is due to competition for molecules of solvation. This is a very simple, salting out is a very simple purification step. So if it works for your protein of interest, it's very nice because it's very easy to do. The issue can be it can denature your protein because, as you can imagine, when you crash a protein out of solution, it could possibly unfold. But for very stable proteins, this works very nicely because it's easy. You can centrifuge out your protein of interest and then move on to the next step. Dialysis and filtration is a method that you can separate things based on size. And so here's an example of a protein solution that is in this membrane here. So if you have a bucket of buffer shown here, and then your protein solution is contained within this dialysis membrane. So for example, your protein are these big blue dots, and then you can have smaller proteins and small molecules that are these smaller dots. This membrane has a specific molecular weight cutoff, so you can buy these in different molecular weight cutoffs, ranging from I think 1,000 Daltons up to 300,000 Daltons or so. And essentially, what you do is you have your buffer out here. You allow this to reach equilibrium, so you have a magnetic stir bar to move the solution around. You let that sit overnight, usually at 4 degrees C. And then your protein of interest stays within that membrane because it can't pass through a molecular weight cutoff. So if you have, for example, your protein is 50 kilodaltons, the membrane cutoff, say, is 10 kilodaltons, and then your protein can't move in and out, whereas the smaller molecules can. So after equilibrium, you purified your protein away from some of these smaller contaminants. Ultrafiltration works in a very similar manner, except for you can do it quicker because you're using centrifugal force. You basically have a membrane sitting here, your protein solution up here, and you can both concentrate and separate big proteins from smaller proteins and other molecules in the same way. It's just you're forcing it through that membrane at a much quicker rate. The last two slides were mainly about how you can do things on larger scales without any sort of resin or chromatography. But by and large, for research purposes and even for protein preparation purposes, people use chromatography. So chromatography is just to purify and concentrate biomolecules based on their chemical and physical differences. So you can do this by size, by exploiting size exclusion chromatography columns. It's also known as gel filtration columns. You can also do this by surface charge, or you can do this by affinity chromatography, and I'll talk through each one of these here. But first, let's talk a little bit about the resin. So the resin itself is based on different matrices. So the resins you can buy from commercial vendors like GE Healthcare, you can get conjugated resins. There's all kinds of different flavors you can buy now, but typically they're some sort of saccharose, agarose, or dextran based. Again, they can come with different ligands on them, different concentrations of ligands. They can have different rigidity to them, different bead size, different pore size. So there's a whole industry built around the chromatography resins. One of the simplest resins to think about and that people use is size exclusion, the resins that use size exclusion chromatography. So in this case, how this resin works is you have your beads, your gel beads from GE Healthcare or wherever you bought them, packed into a column, and you pass your protein solution over it. The protein solution may contain big proteins like these red circles here, as well as smaller proteins and solutes and salts and things like that, denoted by the smaller, I guess, bluish colored open circles. So what happens here are these beads have small channels in them where only smaller molecules can fit through them. And so what happens is when you pour your solution over, large molecules essentially go around the beads and travel down the column more quickly, whereas the smaller molecules have to go into the pores within these beads and basically sort of traveling down like a maze. And it takes them longer to get to the bottom, and so they elute later. And so these resins have been worked out such that you get very nice separation based on size. So your larger proteins elute first, and you can just look at protein elution by absorbance at 280 by, again, exploiting the tryptophan residues as I talked about last lecture. And so you get your larger proteins coming out first, then your smaller proteins coming out, and then eventually your salt comes out. So this is both a way to exchange buffer as well as purify your protein, because you can essentially pre-equilibrate this column in your buffer of interest, put your mesoproteins over, and then your protein comes out first in your new buffer solution. So this is a nice cleanup step that people use a lot in protein purification. Another form that people use is ion exchange chromatography. So instead of size, you are doing this you're purifying your protein based on charge. And so there's two main resins that people use: a cation exchanger. So the cation exchanger binds proteins with a positive charge because the resin itself has a negative charge on it. Whereas the anion exchanger binds proteins with a negative charge because it has a positive charge on the resin. So here's an example here. So if you have a protein mixture that contains albumin, which has a pI of around 4.8, and immunoglobulin, which has a pI of around 8, if this protein mixture were applied to an anion exchanger at pH 7.5, albumin is going to be retained because it has a net negative charge at pH 7.5, whereas IgG will flow through because it has a net positive charge at 7.5. So this is a simple example, but essentially you're separating things based on the PI. So as I mentioned, affinity chromatography is another method that people use, and this is a very widely used method because it's very specific to your protein of interest. So you're exploiting a very specific interaction of the substrate with the ligand. So you get pretty pure product in basically a single step. So it has very high selectivity. You're typically using antibody antigen pairs is used a lot in affinity chromatography. So you can raise an antibody against a particular antigen, and then you can purify that antigen by attaching the antibody to the column itself, or vice versa. You can do it the other way around. Or you can use protein binding partners. So if you know a protein can bind to another protein, you can put one of those as bait on the column and capture the protein of interest. And then you can elute either with high salt or by lowering the pH or by adding some other ligand that competes with that interaction. A good example of that is immunoglobulin G. So immunoglobulins can be purified from blood. You know, you have some upfront steps where you take your blood and you get plasma from the blood, and then you can, clot out the clotting factors to form serum. And if you want to purify the antibodies from that, you can use, if you want to get IgG, particularly from that antibody prep, you can use affinity chromatography by using a column that has recombinant protein A attached to it. So protein A binds to the constant region of the heavy chain of IgG, mainly through hydrophobic interactions. So you can imagine a column here attached to recombinant protein A. You elute over your antibodies, and recombinant protein A binds pretty specifically to IgG, and you can purify away your other proteins that you're not interested in. So here's some real data actually that came out of our lab, that you're looking at absorbance, and if you have a recombinant protein A, you add your sample, a bunch of your proteins elute through, shown by this high absorbance peak. It comes down to baseline, and then you change to an elution buffer, which again can either be lower pH or adding some sort of high salt. You can see a very rapid elution of your, in this case, this peak refers to IgG, as well as some other minor contaminants that are left over. But you can elute your protein, then you can actually reform the column, and then you can run this process over and over again. So just because we have our protein of interest in our elution buffer, we may need to do other things with it for various pharmaceutical preparations or whatever you're using your protein for. So you may need to buffer exchange it into some sort of storage buffer. You may need to concentrate it. You may need to do just the pH. You may need to formulate it with some sort of stabilizer, or you may need to lyophilize it, for example, and we'll talk through a couple of examples there. First, buffer exchange and concentration. So there's a couple of ways to do that. Tangential flow filtration uses a peristaltic pump. Has low shear on the protein, so this is a good method to use. You can also use something called a centricon, which is a commercially available, basically uses a gravitational force. It spins and concentrates your protein at the same time. So you can have to be careful here. You can get some precipitated protein if you concentrate it too much. You can also use something called an Amino stir cell, which is sort of an in-between. It uses nitrogen gas flow and a stirring motion. It's shown down here at the bottom. So it prevents some of that precipitation from occurring. But all these use some sort of membrane that doesn't allow your protein of interest to cross over a molecular weight cutoff membrane, whereas small molecules like water and salts can pass over that through that membrane. Now that we have purified the protein, let's talk about some of the analysis of proteins. Electrophoresis being one of the oldest forms of analyzing proteins. So this looks at the migration of ions in an electric field. It's a very fast, easy, and cheap separation method. So if you have what you think is a pure prep or even a dirty preparation of proteins, you can separate out proteins to look at the different proteins that are in your preparation. This is usually used for small scale characterization, so it's not really meant to recover your sample. It's just for analysis, essentially. But the migration of ions in this electric field depend on both the charge of the molecules as well as the frictional coefficient. This frictional coefficient depends on the size of your protein, the shape, and the solution viscosity. So here's what gel electrophoresis looks like. You have this upper stacking gel shown here, where this has large pores and a glycine buffer, has a lower pH than the running gel or the resolving gel, which is shown here. So these have small pores and a buffer with a higher pH. This stacking gel generates nice sharp bands as they move through the electric field. So the glycines are neutralized in the stacking gel. They only have a small net charge. So this is why this works pretty well. What happens is you get this local electric field that increases and concentrates the sample, and then in the stacking gel, excuse me, and then once it ends in the running gel, it's constant, and you can separate things based on charge and size. If you use SDS, or sodium dodecyl sulfate, you basically can mask the charges on all the proteins, so then you're essentially just separating things based on charge, based on size alone. So this is the case shown here of a couple of SDS page gels. So this is an example of a reducing gel where you take your sample and reduce it so you can lose any sort of quaternary structure. And this is an antibody preparation, which you can see the heavy chain migrates around 55 kilodaltons and the light chain migrates around 25 kilodaltons. So typically the way you run these, you have a molecular weight marker, which is just a prepackaged mixture of proteins with known molecular weights. And then you can run your purification steps along here, for example. So here's an example where they did that, here's a molecular weight marker, here's a pure prep, and then after purification step 1, 2, 3, and so on. And I'll give another example here in a minute. But it's essentially, again, if you use denaturing conditions under sodium with SDS, SDS is negatively charged and it masks the charges on all your proteins. So again, you're just separating based on charge. So isoelectric focusing separates proteins based on their basis of their PI. So sometimes you'll see two-dimensional gels. This is kind of going out of favor now because of the use of mass spectrometry, which I'll talk about in a minute. But if you focus on the horizontal direction first, you can use isoelectric focusing. So the gel itself has a pH gradient, and so a protein that is in a pH region below its PI will migrate towards the negatively charged electrode until it stops at the PI. So basically it focuses it to its isoelectric point. So you can separate things based on this PI in this direction, and then if you want to run a 2D gel, you can actually flip this gel and then run SDS page, so then you get separation both by PI and then by size. So something, for example, down here, has a PI of around 7 and is very small, whereas something up here has a PI, say around 5-ish, and is much, much larger. As I mentioned, this is the 2D gels has kind of gone out of favor a little bit because people are using mass spectrometry, which mass spectrometry is getting better and better. So when we talk about characterizing proteins, a lot of times what people use is mass spec. This started previously by using a much simpler method that I'll talk about here in a minute. But This is used when you have a protein or a mixture of proteins and you want to determine the protein sequence. So if you want to identify proteins in your sample, not just by what size they are or what their isoelectric point is, but if you really want to know what the exact sequence is, these are the rough steps. You'd extract your protein, you might do some sort of chromatography. You could do SDS page if you wanted. You could actually pick that piece off the gel and then run the mass spec, or you could just take the solution and bypass it. But it gets to a point where you have your protein and you can do sequencing. And again, you can do this in a couple of different ways. One is you can just do simple amino acid analysis. So if you have a pure protein preparation, you can look at dehydrolyze it into amino acids, and then you can just say what's the composition, but that doesn't give you what the exact linear sequence is. People have used in a long time, it's been in practice for a fairly long time now, N-terminal sequencing. You can get partial sequence information. I'll talk about that here in a second. But then again, if you want full sequence information, you can use mass spectrometry, or otherwise known as proteomics when you're talking about proteins. You can get up to full sequence information. I'll talk about that. First, I'll talk about amino acid composition. So again, you're just taking your protein prep and in pretty harsh conditions, 6 molar HCl for 110 degrees for 1 to 2 days. You destroy your protein into its amino acid building blocks, and then you just separate those by chromatography. And then you just look at either fluorescence or some sort of optical readout where you can measure the proteins. Then you can run this against standards to where you know, hey, I know glycine elutes here, alanine elutes here. So you can get a sense for composition of your protein. So again, this tells you what amino acids make up your protein, but it doesn't tell you what order those amino acids are in. To do that, again, you can use Edmond degradation or N-terminal sequencing. And how this works is you have your protein and you basically, one at a time, protect the end amino acid and hydrolyze those off one at a time, and then you do amino acid analysis. So this gives you at the end terminus what the first amino acid is, then the second round, the second amino acid, and third, and so on. But you need a decent amount of it, and you can't get through the entire protein just due to losses at each step of the process. So the current state of the art is really the using mass spectrometry, and there's different mass spectrometers that you can use. Typically, one of the main ways people use it is you have an upfront liquid chromatography step. So essentially you take your protein of interest, you digest it using some sort of enzyme. Typically it's a trypsin enzyme that cleaves after lysines and arginines. So you make positively charged peptides, short peptide sequences. You can separate those peptides using chromatography, anywhere from a quick run to 15 minutes or more complicated preps. People run these for 24 hours even, and you can get really high sensitivity. And basically what happens at each time, you can get the parent ion masses for that peptide, and then you can bombard that peptide with electrons, and you basically cut that peptide into smaller pieces using those electrons within the mass spectrometer, and then you get fragment ions. And so basically you can put those fragment ions together to piece together the amino acid, the peptide sequence. And so, for example, here's the peptide sequence of one particular peptide in that protein. You can see that the trypsin enzyme in this case cut after that arginine and after this lysine. And then here's what the fragment ion pattern looks like. And so you can do this for several peptides in the protein, depending on how good your sample is, how much protein you have. You can get pretty good coverage of this. You can mix and match enzymes instead of trypsin. You can get other enzymes that cut after different amino acids, and you can get full coverage of a protein doing it by mixing in different proteases essentially. So it used to be you have to be able to interpret this manually, but now there are software algorithms where you essentially just run your protein of interest, put this into a software algorithm, and then you search this against a database of protein sequences that you think should be in your sample. And then here's an example of a mascot, which is a software that's used in this field. And then it tells you, okay, I think your protein is haptoglobin, for example, based on the peptides that are in there. Then you can go in and click on it, and it'll say, here's the evidence that this peptide, and here's the spectra to back it up. So this is really the state of the art, and mass spectrometers are getting better and better and more sensitive. So this is really the way to go when you want to analyze proteins in terms of what its primary amino acid structure is. You can even get post-translational modifications out of it. So are there certain modified residues, phosphorylation events, things like that. This has become a really powerful technique in biochemistry field. So here's just a few examples. I'd like to talk through, put this into context a little bit. Structural proteins in teeth, some of the main ones you'll find in teeth: enamel. So enamel doesn't contain a collagen like other hard tissues, but it does contain amylogenins, enamelins, and other proteins which are involved in forming a framework for minerals to form. So within this enamel piece of the tooth, even though it doesn't contain collagen, it contains some of these other proteins that form that really hard tissue. The dentin piece contains collagen and glycoproteins to get this tissue in here, whereas the pulp contains collagens, fibronectin, other non-collagenaceous proteins forming this extracellular matrix. So these are some of the structural proteins you find in teeth. Here's an example that's near and dear to me, a protein called LPXD. So this is what I did my graduate work on. So this is an acyl transferase that's involved in lipid A biosynthesis and gram-negative bacteria. It's an antibiotic target because if you knock this protein out, the gram-negatives can't exist. Lipid A is also known as endotoxin. So when people go into septic shock from gram-negative bacteria, it's due to the end product of the lipid A pathway, of which LPXD is an enzyme involved in that pathway. So it's got this trimeric structure shown by the 3 monomers here. This is actually the structure that X-ray crystal structure that we were able to get in our lab. So yeah, it's a homotrimer. This is looking down at the protein. But I wanted to give you this example in terms of the purification of it. So I took this protein and recombinantly expressed it in E. coli, overexpressed it in E. coli, and we got this big whopping band around the right molecular weight. And so you can take this and pass it over a green 19 affinity column. So this column binds to things that bind UDP moieties, which luckily in the case of LPXD, it binds it. Then you can also pass it over anion exchange and purify it even more, and then a final cleanup step using the gel filtration chromatography. So we can get this nice preparation. So this is just an STS page gel. This is the elution profile over the final column. And then you can get nice crystals out of it, so that's where you can get the X-ray crystal structure. So you can also follow a specific activity. So we have an assay for it where we can look at how the specific activity of a protein preparation. So for example, in the crude prep, you get a specific activity of nanomoles of substrate turnover per minute per milligram of protein. And as you purify your protein, your specific activity should go up because more of your protein mixture is your pure protein. So this is what you can see in this case. As you go up, you get higher and higher specific activity, showing that you have more pure protein. And we get pretty good yield as we go through each of the steps. So this is an example in a research field how people use this purification to purify protein and then get the X-ray crystallography, X-ray crystal structure. Okay, that wraps up chapter 2. So I'm going to leave you guys with this study guide here. I'm not going to walk through it, but this study guide is yours to look at some of the take-home messages for this lecture and the previous lecture referring to chapter 2. So take a look at this, and I will see you next time.